Okay, so once again, thank you and greetings to everyone. The work which I am going to present here, the role of metal tolerant bacteria and biochar to mitigate nanoparticle toxicity in barley grown in haplik Chernozem soil. This is a type of soil which found nearby Rostov region, south part of Russia. It's a classification of black soil, so called haplik Chernozem. So the first question which comes to our mind, why we need nanotechnology? As we know, last several decades, that input of chemical pesticides, herbicides, I mean huge input of chemicals in agriculture, damaging the soil, and the same time population increasing rapidly in globally. So to replace, to conserve the soil, conserve to promote sustainable agriculture, we need to change our conventional practices. So for that, we are focusing on nano enhanced product to apply in agriculture. However, before applying these products, we need to know more inside details that how these nano enhanced material is safe for environment. Literally, if we say, if we reduce the size of any material, organic, inorganic, between 1 to 100 nanometer, the material have different properties and different reactivity. So it consider nanomaterial, and these enhanced properties are very useful and undoubtedly last 2 decades. The huge efforts made to synthesize various type of nanomaterial. So how these tiny particles can be helpful to enhance soil productivity and crop production, and how it can reduce the soil stress. Because why I use the term soil stress, inside the soil there are several processes like biogeotransformation, biological processes, microbial processes. So due to a lot of stresses like biotic stress, salinity stress, these processes hampered. Drought stress is also one of the big problem for improving or growing more, taking high yield. Salinity and coping up metal stress. Metal stress is rapidly increasing in various countries due to rapid industrialization, anthropogenic activities. Even here in Russia, there are some big power plants which has released a large amount of metals which polluted even big area of cultivated land. For restoring our soil health and enhancing sustainable crop production, we supposed to, or we must improve the soil, and for that we are applying like bioremediation and phytoremediation approaches last 4 decades, and we are working, but results are not efficient, not sustainable, not durable. So for that we apply nanoparticle to enhance material, so-called biochar. Biochar is paralyzed or made up by agriculture wastage or any other material on high temperature. So this biochar have novel properties also to improve soil health, to provide nutrient, even reclaim the damaged soil. So we can improve the cycles like carbon cycles or nitrogen cycles, or it also helps to improve the unavailable nutrients in soil. So but more recent, we prepared more fine particles of biochar, so-called nano biochar, and these nano biochar in compare with the biochar have more promising results. Considering all these positive impact of nano material, especially in agriculture, there are concern because these tiny particles, when it go to the plant system or in soil, it might have more consequences in long term. It might interact with soil, another elements in soil or soil biota, and can form more toxic elements because of different property. So also some experiments we conducted with various agricultural crops. These particles, when grown in high concentration, and then it accumulate high amount of these particles in elemental form in individual crops. In natural condition, not have any clear information, but predicted, I mean, modeling based information indicated that copper and zinc oxide nanomaterial is available in natural environment. So this is the concerning issue which need more inside explanation of nanomaterial. So for that, since 2015, we have started to test various type of nanomaterial in laboratory lab condition, also in field condition, in hydroponic condition, to try to understand how these metal-based nanomaterial, nanoparticles behave inside the soil in longer term and in short term, also how it behave in water condition, hydroponic condition, and then how it translocate from root to top, root to leaf, and leaf to root by a foliar and vessel application. And then we have tried our laboratory fabricated biochar, wood biochar, and also we isolated some microbes, which is heavy metal tolerant microbes that was isolated from polluted site. We screened these microbes for several metals, and they observed the tolerance ability, and then we apply these microbes in soil. Here we consider also metal polluted soil, and also we polluted soil by nanomaterial. In case in environmental condition, when rapid application of nanomaterial continued, and then there are chances then residue or more concentration will be in environment of these tiny particles. So in case if the concentration increase, how it damage to plant, and in what extent it damage, and where it accumulate inside the plant body. So we observe by various modern approaches, and we found that if we grow plant in nano polluted soil, we found higher accumulation than the bulk particle of same nanomaterial. Also inside the inside the cells of leaf and root, subcellular spaces full of these tiny particles, it was observed and found. And also when we applied scanned microscopic analysis to observe the leaf, we found the patches of nanomaterial accumulation. And of course, the traditional method like atomic absorption method, by that we digest the plant samples and then directly observe by atomic absorption. The high concentration was found in plant leaf and root, even in stems. These particles initially affected like root hairs. This is the first target of these particles when the concentration increased, and also it affect the photosynthetic part. When we observe, this is our laboratory made method for understanding the quick state of plant based on fluorescence. And here we also found that these particles reduce the fluorescence and also damage the stomatal density, even the size and opening of stomata. And when we go for to observe the translocation of these particles, it damage the size of vascular bundles. The number of vascular bundle are reduced, but the size is oversized vascular bundle were observed. Also, the similar results were found in leaf system, that nanoparticle translocated from root and reached to the leaf, and it also damaged the leaf cells. Here we go in more detail to understand that where these particles, if the concentration is higher, where it really affect. So we can design our particles, or we can coat nanomaterial so it not express everywhere or express only like a smart delivery. So when we go for the chloroplast, here we found it affected like thylakoids, and also affect the size of chloroplast, starch. So all these subcellular organelles were hampered or damaged by the nanomaterial. We also observed a lot of physiological parameters and a lot of physiological enzymes, biochemical enzymes, and we found if the certain stage like more than 300 or 100 mg per liter or per kilogram of soil, these tiny particles increase, then they damaged or they increased the biochemical enzymes. But when we applied biochar and metal tolerant microbes to overcome this toxic effect, and we found if we see here like this is the clean plants, clean soil, and the root length is almost the similar when we apply is like biochar and bacteria, and also the size of root. So these particles toxicity was elevated or reduced when we used biochar and metal tolerant microbes. And we also going for in more detail to understand the subcellular stories, and it also shows when we apply microbes and nanomaterial, it restore the subcellular organelles, and the functionality of microbes increase, the community of microbes also increase in soil. So also the possibility which we use biologically synthesized nanomaterial, especially for Saunella species mediated biogenic nanomaterial, or plant root isolated in this experiment, like a legume root nodules microorganisms synthesized nanomaterials are more safe or biodegradable for in environment, and that is recommended to use for further. But of course, before using, we need to see, or we need to coat these particles which we are working. I mean, we are trying to coat these particles, so these particles shouldn't express everywhere in plant. It only express either in photosynthetic part or the place where it needed. So in this case, we may reduce the input of these particles. I mean, the less amount of nanomaterial can give maximum results without damaging plant organelles or without damaging environment. So in conclusion, we can say that these particles, of course, because huge positive application, so we can't avoid to include in agriculture, but the challenges associated is still like if metal-based nanomaterial applied in agriculture in soil, then how it behave for 10 year, 20 year, if it is not degradable. So this study we also conducted on microbes. For example, 5 year, we are continue observing the behavior of these metal waste, zinc and copper oxide nanomaterial in soil. every year the toxicity decreasing compared to the previous one. So we can say it might be safe, but still we need some more years to observe the behavior inside soil. So the suggestion is that nanomaterial can be used in agriculture, but long-term explanation is required. In Russia, still we have not adopted in agriculture. So some of the books which published on nanomaterial and its toxicity and safe application in agriculture. So thank you so much for your kind attention. Soil is life, so recommended it should be conserved. Thank you so much.